Introduce yourself.
Right, I'm Piero Carninci. I've been for like 28 years in Japan at RIKEN and running the various functional genomics project, Phantom project. And from the beginning, I've been involved in the human cell atlas from the very first meeting, and we started with Jay and other colleagues, Chung and others, to coordinate the project at first in Japan and then expanding to Asia. And my pleasure to be here and interact with all of you.
Okay. Hello, I'm Shin, and I am currently based in GIS, Genome Institute of Singapore in A-Star, formerly at RIKEN, and been with Human Cell Atlas since 2016-17, so quite some time. And it's great pleasure to be here to talk to you about new phase, the next chapter of Human Cell Atlas almost, as we move into spatial, diversity, and disease. So, we love acronyms. So SDD, if you guys ever want to think about for remembering this, SDD means for spatial, diversity, and disease. But today, here, you're in this room, we walked across this very nice passage, finally got some sunlight, to really talk about disease. And, this is a breakout session, and it's meant to be for you guys and not really for me, and to hear your thoughts when it comes to disease and how to move forward. So honestly, we don't have any slides, and it's meant to be that way. And we have few questions that we want to ask to you, but we really want to hear what you have to say, and also just doesn't have to come up with concrete action items, but we just want to hear what you think. And then maybe you could move forward about what are the necessary steps for collecting samples and asking key biological questions and the technologies and so on. So are there any questions before we start? Yeah.
Maybe we can just remind you that the Human Cell Atlas was started to make at first a map of the healthy human body. And so we've been insisting not to focus on disease sample from the beginning, because we always would need the reference. But of course, we also started to realize that many funding agencies, particularly not only in Asia but in other countries, also require to work on a disease. And I think that's probably this is the right time to start to really think to integrate with a disease atlas, which is going to be massively huge and very complicated because of all the difference in genetic background, type of diseases, and age, and all the possible aspects that affects the human health.
Okay, so as Piero mentioned, the disease, of course, is a spectrum. I think we all can appreciate it's not so binary as we hoped. And, when we say healthy, even healthy can be a very difficult definition to define. And in a certain way, it's also a continuum and spectrum of various molecular and physiological features. So we are starting this new initiative of disease only because now I think with spatial technology, it's becoming a lot more accessible and a lot more feasible. And I think it's the right time with the technology is now really mature or relatively mature that we can really think about expanding the atlas to disease. But of course, we also entering into phase of what are the needs, what are the global needs of understanding disease, but also what are the regional needs. We are here in Asia, so what are some of the prevalent disease that we face, but also what are some of the local needs to address some of the funders that you're reaching out to, because we also want to address our neighbors and our nearby society. And so there are multiple layers of thinking about disease, just even in this scope of context, right? So maybe I just open it up to the audience. Would love to hear from you, since we are from various parts of Asia. Like, what are some of the local disease burdens that you face that are really important and relevant, and that it would also excite your community and the funders to really get you to study this aspect? So I'll really open it up, and just raise your hand and just feel free to speak, and we'll take notes. Yes. Yeah, we can also take notes here. Yeah. Yes.
Hi, I'm Piero from Sri Lanka. So, as we are from Asia, like everyone might have known about dengue, and Sri Lanka is suffering from dengue for like last 4 decades. So yesterday also in the IDA, I have discussed about like how we can move forward as a collaborative project with our neighbors. And I have attended Human Cell Atlas single cell hands-on experience in Thailand this June. And from this point onwards, we know like much better when it comes to pathogen, but when it comes to severity of dengue, we think like host response is needed to be much explored, and we need more understanding, because region to region people's like we are the human genome is different. So how we are going to move forward from here, and how Human Cell Atlas can help us to initiate this and share our data or any possible platform to do that.
Thank you. Anyone else? Martha?
So I will talk more about chronic disease, and I'll talk about two kind of sets of diseases. This is not just one disease, but sets of diseases. What we've been interested in is oral cancer, primarily because of its very high prevalence, #1 killer among men in India, among the cancers. We haven't been able to touch cervical cancer, which is the other among the females, that's the highest prevalence. And of course, single cell studies on cancer are very interesting, especially when it is three-dimensional tissue. So that's something that we might pursue. And this is not just for India, but also for the neighboring countries and other places. And with respect to other places, for example, if you look at the TCGA data, you would find oral cancer is predominantly tongue cancer. When you come to India, it's predominantly, other than tongue cancer, it's the rest of the oral cavity. And we know the reasons why this happens, the way people use tobacco. That may be just one reason, but also it's very interesting to look at, the single cell distribution in the tissue. The second one that I want to talk of is more complex, but it's really becoming a major, it's spreading like wildfire, which is T2D, type 2 diabetes. And type 2 diabetes is essentially an endocrine problem, but it also afflicts other tissues such as the liver. Non-alcoholic fatty liver disease, or now it's been renamed as MASLD, but it's really a continuum, something that you actually emphasized. And so it starts with an endocrine system, but afflicts many other organ systems. So that may be another disease that we would like to look at, or even other people would like to look at, because type 2 diabetes is so prevalent and is becoming more and more prevalent in other regions as well. I'll stop there.
Yeah, I just want to touch upon your last point, that actually in the recent global survey of burden around the world, metabolism and metabolic disorder, such as type 2 diabetes, is one of the biggest burdens that are on the rise compared to other diseases. Anyone else? So we heard dengue. Maybe I'm going to broadly say infectious disease as well, to cancer, specifically oral, to cervical cancer, and you heard type 2 diabetes. Pump up, yeah.
Maybe I'll add a little bit more. We will have a breakout session on infectious disease tomorrow. So anyone interested in, please come join us. So I'll expand to the infectious disease, particularly dengue, that we've been working on and discussed together. And but besides dengue, there's also other important infectious diseases such as tuberculosis, malaria, which are still quite prevalent in our region. And also to expand further on cancer, we also have non-small cell lung cancer that's increased in prevalent in Asian female for unknown reason for now. And also liver cancer, which is also prevalent in our region as well.
In Thailand. Anyone else? Or another question is like, what disease are you working on? Is another question.
Hi, I'm Zhang Xiao from BGI Shenzhen. Actually, I always have one question about the disease, like the senescent cell related disease, because senescent cell people talk about most of the time people talk about this at aging process, but for this year some degeneration disease like AD, Alzheimer's disease or PD. People see that in brain or in other organs, senescent cell will be appear in different cell type like microglia and also other cell type. And these cell type secrete some SASP molecules to affect other cell type and other organs through blood or other things. So I want to know how people like thinking that senescent cells, that's really these cells exist. People use beta-gal staining or gamma H2X staining to prove these cells exist, but different study they have different conclusions for this. That's my questions.
I'm not an expert in aging or senescence, so I don't know if there's anyone in the audience want to comment on how to characterize senescent cells.
Yeah.
Because I think that's been a question for a long time.
Or like spatial. I think that
Using
latest
spatial.
paper on cells from Liu Guanghui lab, they do the aging mouse that like doing the hotspot for senescence.
Yeah. I know that from a genetics point of view, such as in the brain, there are more somatic mutations that are occurring in glial cells than neuronal cells. And they're suggesting that this acquisition of new mutations over age can lead to alterations in cellular physiology. So whether those are linked or not, I'm not sure, but it's something for us to explore. How age or clonal hematopoiesis or age-acquired mutations can lead to disease burdens. Yes.
I want to kind of point out, aging is not disease. Aging is the status of the cell. If you try to understand the cell status, right, aging is not disease, right? Are we going to discuss something is not disease or something is disease? That's need to be clarified, right? So.
Yes, I love this discussion. There was no right answer, but yes, age is a process. I guess age-acquired disease or age-onset disease, maybe it's fair to say. But whether it is due to mutation, or whether it's due to epigenetics, but these are very important questions.
I think aging. I mean, of course, I will say probably, if you go into aging, we probably need to really think at profiling chromatin status, epigenome status, and also all the spectrum of the mutations. So I think that we probably need to shift gear into much more functional aspect of how I would say probably a lot on the chromatin level, and we need it. Okay.
Yeah.
Yeah. Sure.
Yeah. Telomere shortening. Both, yeah.
So I think Partha already mentioned type 2 diabetes, which is a huge burden, and also associated diseases, cardiometabolic traits. So for example, coronary artery disease and, cardiac fibrosis, things like that. But also like from a tissue collection standpoint, I'm not sure if this has been brought up yet. Blood is PBMCs is easiest to collect, and anything that's associated with inflammation, like autoimmune disease and immune-related diseases. So just to name a few, maybe asthma, Graves' disease, rheumatoid arthritis, lupus, all these diseases can be immediately studied with the data that we have on hand. So if we're trying to, gather data from all around the world, I think PBMC is still one of the easiest to collect, especially in low resource setting where maybe they don't have access to, more advanced medical equipment to collect other tissues, right?
Absolutely. Yeah.
Where there are collections, but do we have single cell data on those? Or are they even amenable to single cell analysis at this stage, even from the repositories? No, for asthma and all of the PBMCs that you talked about, I don't think people have actually generated single cell data and they've kept it in their refrigerators. But once you bring them out, I'm not very sure that they'll be amenable to single cell analysis. I'm not sure. It may be, but I doubt that they will be.
So in my experience, I'm not sure about other parts of the world, but in Singapore, there are doctors that are banking, for example, sputum, or like the synovial fluid, but they're in the banks. But these doctors are not single cell specialists, so they don't even know where to look. So then for me, I feel like it's my responsibility to reach out to them and to say, you're sitting on a gold mine here, and let's collaborate, right?
Yeah, there's a hand over here.
Hi, I'm a rheumatologist. We want to know more about the rheumatology disease such as lupus, arthritis, and many of the autoimmune disease. But now there is amount of single cell data in cancers, but very little data in the rheumatology disease. We did some single cell data, but we found the PBMC cell types have very little changes between the autoimmune between the rheumatology disease and the healthy. So we want to know more about this.
Yeah, that's a really good point. I actually wanted to ask you, do we need, what kind of organs or tissues will be most relevant to profile immune cells in? Because I understand the systematic blood will be very different than tissue resident blood. And in the case of disease, that would also be, I guess you're saying, could be very different. So in your case, which organ will be most interesting, but also feasible in studying rheumatology disease or autoimmune disease?
PBMC.
Just PBMCs are sufficient.
Yes, because that's the lung tissues is difficult to take.
Yeah. So if you were able to, so my question is there added value of doing tissue extraction to look at immune cells, or is PBMCs sufficient to understand the characteristic of the disease? Maybe, yeah.
Because the rheumatology disease affect many organs, and it's difficult for us to take the tissues such as the lungs, the kidney. For us, take the PBMC is the best sample.
Okay.
Yes, but we can't find too much difference between the rheumatology disease to the healthy people. We want to know more.
So that's my, yeah. We're like, then do we just sequence more patients, or do we need to look somewhere else?
Yeah. I'm not sure. Yeah.
I don't know. I mean, for example, should we look at secondary lymphoid organs like the spleen, or should we look at lymph nodes or tonsils? These are organs that where immune cells are extremely activated and even perturbed, and that's can lead to autoimmunity or other, collateral damage to your whole system. So if you could think outside of systematic blood that can help to better characterize disease, I guess, would those organs be useful? Characterizing those organs be useful? Tissues, yeah? Okay. I'm just throwing it out there because again, I say this, I understand tissue collection is extremely challenging, but there are some tissue banks or clinicians who have some FFPE sections of some of these tissues that we're talking about. So question is whether we can also apply spatial technology for some of these clinical samples. So if we can characterize immune cells in the spleen of a lupus patient, would that be useful? Would that give you a better understanding of the disease than just looking at PBMCs, or we look at it together or something like that? Yeah. I will just, for the open discussion here, just wanted to see what kind of added value we would have if we were to introduce the spatial technology in addition to, say, PBMCs of different organs. Yeah. Yep. There's a point, there's a hand behind you, maybe Pompan can answer.
Maybe,
Yeah, but we'll continue this discussion. We don't have the answers yet. I'm just throwing in a question.
Just chime in a little bit. So I was thinking, for example, some autoimmune disease has a lot of arthritis. Maybe easy tissue sample that's locally could be synovial fluid. And we can profile immune cells in the synovial fluid. That's quite easy, doesn't require biopsy. Or in certain diseases such as SLE with glomerulonephritis, you can potentially, if we do spatial on the kidney, might be helpful.
Mhm.
I would think it's just ask rheumatologist someone.
Yeah.
I'm an allergist, by the way. So in asthma that Bo-Sien mentioned, maybe there's some already profile in the lung atlas and then human asthma lung as well. And might add some other systemic allergic reaction and allergic rhinitis, which seems to be mild, but it's very common, up to 20 to 50% of population. And the sample is quite easy to collect for nasal swab or even nasal polyps that people sometimes cut it out. So
Yeah.
that could be potential tissue source.
Yeah. Maybe I'll also shift the question a little bit differently for kind of different topic. So Bo-Sien raised that there are some tissue banks or clinicians nearby that sometimes you just have to make that extra step and introduce ourselves and say, hey, we have this amazing technology, single cell genomics and spatial. Could you provide your tissues for analysis? And when I was in Japan, I had interaction with Brain Bank, and they had 500 postmortem brain tissues, and they were begging me to use them because nobody was using their brain tissues. I was like, I'll be more than happy to. I just don't have enough money. 500 tissue samples out, and this is human brain, which is multiple regions, and you will require a lot of profiling. So, sorry, maybe I'll finish this question is, do you guys have access to such tissue banks, or do you guys have access to known clinicians who could provide potential? It could be fresh frozen, it could be FFPE, and that you could really scale to a point where it could be part of the Atlas efforts. So I just want to pose that question and hear your thoughts.
Hi, I'm Siglu from University Malaysia. So in our faculty, we have this cell mentor program. So this is apart from collecting samples from the clinical side from patient donors, we also work on the tissue samples that is accessible from the cell mentor program. So it's a body donation program for research and medical teaching purposes. Yeah, so these are majority of them are elderly donors, but sometimes we also have younger subjects. So recently we have the donors who are as young as like 20 years old. Yeah, so add on to just now about aging. So I think if talking about aging, I think osteoarthritis or some of these musculoskeletal related degenerative joint disease shall be looked into. For example, osteoarthritis and yeah, because until now there is no markers to diagnose it, or there is no any treatment to reverse it. The only treatment is total knee replacement at the end stage of the osteoarthritis. And apart from that, about just now, I think someone mentioned about metabolic syndrome related. Yeah. So I think systemic low-grade inflammations is also associated with a lot of diseases, including these osteoarthritis. And so I guess it would be good to profile some of these cells associated with the systemic low-grade inflammations. Also in relations to the degenerative related joint disease.
Yep.
Yeah.
Did you want to say something?
No,
Okay.
some additional
Yep.
like,
Yes. And
Hi, so I'm Alex Sharland from Sydney, Australia. So I guess we have one foot in Asia and one foot outside. I work in transplantation. I wanted to come back to the issue of whether you really need to sample the organ that you're interested in to look at an organ-specific immune response, or if the peripheral blood is good enough. And I think we don't know yet.
Correct.
We're trying to do both. There are certainly limitations on collecting human tissues. And in terms of repeated biopsies, patients hate it. It has morbidity, sometimes even mortality. Everyone wants to move to less invasive diagnosis. So one thing that's happened in our field is people are thinking about, well, what is a substitute that's not the same as, say, a kidney transplant biopsy, but better than peripheral blood? So people are looking at sentinel tissues, sometimes putting a skin graft in place that could be biopsied less invasively than, say, a lung transplant, or, you know, something like a pancreas that you really can't access.
Yep.
And, you know, perhaps there are even better alternatives to that. So organoids, for instance, might be a better sentinel than a different tissue.
correct. Yep. That was something I was gonna bring it up, but yes, excellent point.
Right, if you mentioned about organoids, there's a comments regarding the immune cells and organoids. So there's a huge difference if you do a single cell analysis between the organoids in your lab, or if you're talking about kidney, right? I'm a kidney person. If you make a kidney organoids, and even Melissa Little in Australia, right? Her single cell data, if you compare that with the actual fetal kidney, it's completely different. Doesn't matter how careful you do, we add flow, we add the macrophages. It's not been published, right? But still, the thing is phenomenally different. You know, by look at the cells, if you make a good organoids, the tubule cells are actually smaller. You know, they have more number of cells to form the tubule, but the actual fetals, the tubule cells larger, and they use less cells to form a tubule. So this is completely structure, biologically, and the ISEC level, they're all different, right? Sometimes they jump the developmental stages from S-body into common phase. It's just they don't have that in organoids. So you might study some of that using organoids, but not the entire process. However, we use that to validate our data. If you have a lot of data-driven based research and you don't know what's happening between the different time lapse Organoids can be the tools to validate your cell types. Organoids can be the tool to validate to your different calculations. People talk about the different kinds of, computation approaches, but whoever going to validate all that? Organoids can be the tool for that. So it's just different things with different use. Thank you.
Peptide rectifier of the HLA, which differs from one cell type to another. So perhaps an organoid might recapitulate that more closely than a different type of tissue.
So in the same vein, I will also mention that again, we don't have access to too much of funds, so sample sizes are not great, then equal to 1 essentially. But you mentioned, and Boxiam mentioned that, reach out to people who have tissue banks and so on. We need to be very careful. We've done N equal to 1, fresh, frozen, fresh, and frozen FFP block. Very different results.
It is spatial or single cell?
Single cell.
Oh, yes. Yes.
Very different results,
Correct.
RNA-seq. So one needs to be careful. Just because somebody has a repository of tissue, tissues in the bank, we just pull them out and get some data. They may not actually replicate what we could get from fresh, samples.
Absolutely. And some of these are also technology dependent as well.
Yes, of course.
So yeah, which we'll definitely need to discuss. I guess one of the motivation here today is to also look for any level of consensus or synergy in a way that we can actually galvanize some key ideas so that we can reach out to funders who may be interested in some of these topics. So yes, right now it's just want to hear your thoughts openly, but maybe over time we can gradually move towards certain common goals, and then we can create flagships around that. But again, we don't have to do it today per se, but just want to hear what's out there in the community.
Yeah, I want to make one more sentence.
Yeah.
As we are talking about disease, we need to think about study designs and sample sizes much more than normal.
Absolutely.
So we also need to keep that in mind.
Yeah, absolutely. So I actually want to plug in tomorrow. There's a breakout session where I'll be co-chairing the Skin Atlas. And the reason why is, I did my PhD in a dermatology lab, but skin is one of those very relatively accessible tissues that can be acquired. But to Partha's point, even atopic dermatitis can have a huge range in terms of its severity of disease. So then do we need 50 samples for mild and 50 samples for severe? Or how do you stratify this? Where do you demarcate the boundaries? So these are definitely questions that we need to address, and how many control samples that we would need to really address, sex matched, age matched, ethnicity matched. All of these are extremely key questions that we need to address. Gender matched. Yeah. So, so scale will matter. I think in order to make a very robust atlas, we need scale, we need diversity, and we need you guys to, provide access to these samples so that we can really integrate some of these data together and make a unifying atlas within Asia. So that's, again, kind of part of the motivation why we are meeting today. Yes.
So because I think that the scale, as Partha said, the scale is going to be very, very important. So because it is difficult to really do so many diseases at a very large scale, shall this group discuss if we need perhaps to focus on feasible, a certain number, limited number of diseases to really go for a pilot to sequence, profile, whatever is the number across several population, and having a pilot study like this, and see what we learn, and face other diseases later. And
Absolutely.
Starting from the most important. Yeah, Jonah.
Yeah, just kind of on this point. So in, I think it was Aviv's slides this morning, she was kind of noting that there is this adjacent, disease networks, Helmsley and IBD and Crohn's, Human Tumor Atlas Network, there's a
KPMG.
senescence network, KPMP. How have they answered that question? Right, like they've been working on this for some number of years, and
Correct.
do you have a sense for, how much coalescence or how much structure or lack thereof they've applied to?
Yeah, that's a really good point. And in fact, a lot of these consortiums are centered around the tissue, right? So KPMP is kidney specific. So I think they're looking at 2-3 disease cases of the kidney, chronic disease of kidney. And I think Hemsley is probably more gut centric.
And IBD.
IBD. Right. Yeah. And so in a way, do we want to create such effort in consortium within HCA that will allow us to go deep in one of these tissues? Or do we want to spread across many diseases, which I think it could be risky, but it's also a possibility. I don't have the answer. Yeah. I think a lot of these organ-specific consortiums are, at least I know from KPMP, they are quite successful in generating data, but largely from the US. And they're entering into a lot of spatial data generation as well. IBD, yeah, in Hemsley, yeah. How's Hemsley? Is it I know it's a funder, but do they also have a consortium around?
Yeah,
IBD?
they have the Gut Salalis Consortium that had a kind of normal reference, but I think the way that you're discussing some of this, there's also a very deliberate focus on clinical samples and Crohn's and IBD.
I see. So that's a really interesting point, because we do have 18 biological networks in Human Cell Atlas, which represents various organs. And obviously, we don't want to do this alone. In fact, the disease that we will select would have to be closely linked with these key biological networks and be able to use their expertise of referencing and how to build the disease atlas beyond that platform, because we don't want to reinvent the wheel, right? Yes. So.
Yeah, I just wanted to suggest like pneumonia as a particular disease. It's very common across like multiple different countries, caused by several pathogens. It's easily accessible in the sense that you can do nasal swabs and be able to look for it. And potentially within that, you would get information both from perspective of various diseases, but also if you think about like environmental stressors such as pollution levels in different countries could be different. And so that could be a easily accessible tissue to look into.
That's a good point. And I think related to allergy and related to even infectious, like COVID, for example, or other flu, yeah, the nasal swabs, I think there are not only immune cells, but a lot of epithelial cells can be also acquired. And I believe Jose, someone in Boston, yes. So there are actually a standardized protocol, or at least protocol that we can adopt within the community to standardize how we can collect nasal samples. And in fact, this could be a great way to also expand diversity and study disease at the same time. That's a good point. Yeah. I know we talk a lot about spatial, but yeah, it doesn't have to be. Oh.
Okay.
Yeah. So what can we learn from other disease specific consortiums? Yeah.
And there also we could find some biological network that
Yeah, absolutely. I'm just Esther. I'm just being mindful. When do we? Oh, okay. So we have 15 minutes. Plenty of time.
Is there? I mean, years ago, you guys started to organize the ADA project and started to really get it together, to really create a consortium across Asia and discussing priorities. And PBMC was a very easy and obvious sample. But shall we see if there is any possibility here in this group to identify some of the diseases that would be helpful for to attract funding in your countries, and see if we can really converge once more in some initiative. I mean, I can easily see this cancer from one side, some type of cancer, and for sure some inflammation, immunity related are of course infections as well. Is there any? we are not going to solve the problem in 12 minutes, okay? But if we could probably start to know if anyone has any idea how to or any additional comment on this.
Yes.
Yeah. I mean, I'm not a funder, but I know there's funders in the room. But so I think it depends on whether we're attracting central funding from the CZI or national funding from our respective institutions. But if we're trying to attract central funding from CZI, what are the criteria that they're looking for? Number of people with that disease, severity of that disease, easiness of obtaining samples. I don't know, maybe there's more, but I cannot think of it off the top of my head, but we can brainstorm what are the criteria. If we were to come as a whole, as a community-wide effort in CZI, what are the key criteria to rank this disease and score this disease? And then, I think we have a clear picture.
You can ask the gentleman sitting behind you, Jonah, from CZI. We'd love to hear from you, Jonah. I know, yeah.
Sure. So I'll talk a little bit about the way that myself and my group and some others are thinking about disease in my talk. So maybe to bury the lead a little bit there. Maybe one thing just to touch on is I think one of the reasons that we have supported so many of the projects that you've heard about today and others is we're not really a funder of disease. Like that's not the primary thing that we do. We think about basic science and technologies. Now, I think a real success or win for us, and I think for many of you, would be for some of those technologies and those kind of early stage references to spin off and start to be used and by extension supported by many others. The caveat to that would be examples of disease where from our perspective, we're not learning about a disease, but we learn about classes of disease or ideally all disease, right? So that is like at the moment, we're really focused on this kind of basic question. So I think there are some instances of that you all are touching on, something like inflammation. Again, I'll come back to this as kind of one of those areas that is not, it's a broad class, cause or consequence, implicated in infectious disease and cancer and many others. So that is something that we would be interested in. But I think that the real opportunity for many of you is also kind of using that platform, using some of these core resources to start to think about other funders that have these really clear remits. One of the reasons that our founders didn't have a disease remit is they kind of looked around and understood that that's the way most science is funded. So we try to complement that and do something a little bit different.
Yeah. So you did mention Hemsley or really focus on.
It's a great example. Yeah. Like they would not have supported the gut cell atlas if it weren't for the fact. Like I don't think their trustees would have approved it if it didn't have an IBD and Crohn's.
Correct.
Can't speak for them. But
or Gates Foundation in case of infectious disease in some scenarios. Yes. Yeah, so I think these are also opportunities for us to seek and what other foundations that could really support some of the disease we listed. But also, I think a lot of local funders are, especially in Singapore and Japan, cancer obviously is very dominant, but also at the same time very competitive. But we should not always shy away from the big challenges. So yeah, cancer is obviously something that can bring, but there are also other cancer consortiums that we can learn from. But there are also cancer types that are far more prevalent here in Asia than Western countries. You heard of oral cancer, but also liver, gut, gastric cancer, things like that are heavily associated or highly relevant here in Asia as well. So that's another angle that we can consider for HCA. Yeah. Anything else?
I'm wondering, sorry, just to brainstorm, but there are some countries, as you mentioned, that would be able to support, I say, Japan with AMED would be able to support specific diseases, and Singapore, as you know very well, would support probably other countries. And there are some other countries that where the funding is not going to be enough. Okay, so and what is a possible strategy for supporting countries where funding is not going to be available? Gates Foundation or anything? I mean, is there any strategy that we should discuss here too for that? I mean,
usually
we are here to get ideas. We don't have.
Non-government organization will be more willing to fund this type of.
Yeah. For the Gates Foundation, I mean, we have a fund from the Gates Foundation, but that was really targeted towards vaccine efficacy, because we know that, like, the immune responses are very dependent on population. And Gates's major focus so far has been vaccine research. So like they're sometimes very interested in like, let's say, design for a particular vaccine where there are no very clear correlates of protection. And if something like those could be identified through single cell sequencing, that could reduce the size of the trials that they have to do to come up with showing that, oh, this is efficacious versus not, would potentially be something that they might be interested in. So, I mean, like that will also speak towards a more translational work in helping make these things a little cheaper.
It's a really good point, and the word translation reminded me for us to really also be open to industry collaboration. And I think there are opportunities where if we as a community come together and say we have samples from India, Singapore, Thailand, Vietnam, Japan, Korea, China, and that we are actually building an atlas using common platform or strategies to do this, to study this particular disease. I think it's really extremely attractive for industry to support this kind of effort in some ways. And it's just for us to at least keep that in mind. We may not necessarily have a concrete company in mind, but the amount of resource that we can collect and build together as a community is something that no single lab or no single country can do. And that's where I think HCA can really shine, is this type of collaboration and community-based network will be very, very powerful. Yes.
I wanted to just come back to thinking about liver cancer, because one thing about it is that, you have this convergence of initial insults that could be infectious diseases, could be metabolic disease, alcohol, even autoimmune disease, and they all seem to end up passing through this stage of fibrosis and inflammation and ending up in carcinogenesis. So looking at how those different pathways converge on the one endpoint and what are the similarities and what are the differences could be interesting, and because of the different pathways could allow involvement of multiple groups from different discipline areas.
And there'll be surely a lot of clinical samples that are available for that. So I believe there's a cancer breakout session tomorrow, just to plug in that aspect as well. So if any of you are interested in building cancer atlas, perhaps could be a good opportunity. And you're absolutely right. And liver is one of the prominent cancers in Asia, along with gastric and colon and so on. So it'll be very interesting to study that as well. And maybe there are some industry funding for there as well. Okay.
Most of the time, ask for immediate translation of the research. If they don't see immediate translation of the research, they are usually, at least most funders, most industry funders in India, and I suppose that's true for many other places of the world also, they need quick return for the money that they will invest.
Yeah, we will, if you are at that stage, I think we will need to write down what are the terms and conditions, because I think it really depends on how we formulate this. But you're absolutely right. It will be definitely closer to more of an application rather than fundamental science for sure. Yeah. Yes. Well, we have 3 minutes. Yes.
Okay, I will be quick. And this is Chen Wei, and I'm currently a PhD student in China. And what I wanted to raise the discussion is that what our purpose to construct the disease atlas actually for the ultimate goal, it may be to find some new treatment or some new drug to cure the disease. But currently, I found that lots of the disease atlas is just if it have functions, it's more likely to raise some new diagnosis, but we still have a long way to find out the treatment. If there is a way to find out treatment, and I think it will be much easier to attract the industry to funding to construct this kind of atlas.
Absolutely.
Yes. So I just wonder what is still need to done to let the disease atlas to help us to find out, especially some new drug. Yeah.
Yeah. Yes. So how to?
So I just quickly comment on this. So because we know the AI models being hugely used in this kind of application, is about a Nature Biotech paper there and a few other papers coming out. They basically use the different kinds of ISEC data. They analyze different status of the cells, and then they can actually narrow down different kind of pathways. So they might not give you specific drugs for that, but they certainly give you targets for that. Then you can switch to another model to calculate what the targets for that kind of chemicals, how they achieved. Then you can simulate those kind of compounds, and they use the automation to validate your things, your organoids. So I think it's somewhat in the nature, they try to have a review out quickly. So to turn over this kind of, you generated lots of data, how the data being generated, how's the data being used, what kind of different model, transfer model or diffusion model, whatever model they have to generate different targets, and targets related to compound to validate it to do a circle. So I think very quickly people will have the total solution for using this data to achieve some valid treatments.
Absolutely. And I think now there are additional drug modalities, not just chemicals, but you can argue antibodies or biologics to nucleic acids to cell-based therapeutics. So yeah, I think these are all possibilities. We don't know exactly how it will pan out, but you're absolutely right that we can start with target discovery, but then with possibility of additional drug modalities that could also entice some of these industries to engage with us. Yeah. Maybe we should close. I think it was very fruitful. I know it wasn't very concrete, but I think we got a lot of feedback from you, and hopefully it's been useful to think about what really interests you as well, because you probably heard many different ideas. And like I said, in AIDA, I started just by having this kind of conversation, and we just got together. I heard you talk about immune cells, and I heard you have this technology. Can we collaborate? And then that's how AIDA started. So I hope you guys got to know each other a little bit through some of the comments that you guys made, and you guys can interlink and maybe start some flagship projects amongst yourselves. So thank you. Do you want to any last comments?
Yeah, thank you, Jay. I think that this is very important. This is very important. It was a very short time. We didn't discuss about particular technologies that we should use. Shall we use profiling, long read to capture RNAs that may do something, or chromatin status and spatial versus many more approaches. I think that this is just the beginning, but of course, if you have ideas on on specific approaches, please do not hesitate to contact us. We can construct around that. Thank you.
Okay, thank you very much.